Well, folks, this is the lecture you've all been waiting for. Today I talk with you about photosynthesis. I say that a little bit sarcastically, but we're going to have fun and do this last sort of biochemical lecture before we really get into the biology, because I got good news. On Thursday, for the first time, I started to really talk with you about cell division and how all of that fun stuff with DNA works. But one last lecture that's sort of rooted in chemistry, and then we'll get into the stuff that really gets me excited. Just wait till you see how animated I get for those types of discussions. But again, we're rounding things out today. I'm going to talk with you about photosynthesis. It's sort of the yin of the yang for metabolism. The last two lectures I've talked with you about catabolic processes, how it is that in metabolism we break things down, and specifically where we're talking about how we break sugars down so that we can extract energy from them, releasing CO2 and water along the way. But today I talk with you about where those sugars come from in the first place. And so this is anabolism. We're building things up. Today I'm going to talk with you about how living things capture energy and store it in covalent bonds that can then be tapped into later on through those catabolic processes that I've talked with you about the last two times we've met. So again, today, bottom line, we're talking about photosynthesis, and all of that happens in the chloroplasts, so I'm going to be showing you a lot of green pictures today as we talk about how the structures of chloroplasts end up allowing us to do this trick, or allow living things to do this trick. Before I go there, let me just toss out one pair of vocabulary words for you. These are in the lecture notes. I think it's worthwhile for us to at least have contemplated them for a little bit. There's two different kinds of organisms that you might talk about. You can divide organisms into two neat categories. There are the autotrophs and there are the heterotrophs. All right? You and I, we are hardcore heterotrophs. What that means is that we have to get our energy by stealing it from something else that had collected it and harvested it and stored it in covalent bonds that we can then use to our own advantage. So a heterotrophic organism is one that is not self-sufficient in terms of energy. It needs to go elsewhere to take energy-rich molecules to be able to drive its own metabolic processes. In the contrast, the other category of living things are the autotrophs. Need I say more? Autotrophs they can be self-sufficient. They don't need to take their energy from other things. Now, that doesn't mean that they can't. It just means that they don't have to. Right? So plants, for instance, are an exceptionally good example of an autotroph. They harvest energy from sunlight and store that energy then in covalent bonds that heterotrophs later on can come and take from them, or that they can use for their own purposes if they're lucky enough to not be eaten by some heterotroph. There are two different kinds of autotrophs. There are the photoautotrophs, the things that capture energy from light, like plants do. And then there's a less often talked about category, the chemoautotrophs, that harvest energy from energy-rich molecules, from chemicals that they find in their environment. And it would be cheating if those energy-rich molecules were sugars that happen to be floating around. When we're talking about chemoautotrophs, we're talking primarily about organisms that live in deep sea vents, high pressure, high temperature, where they're getting their nutrition not from sunlight, where they're getting their energy, I should say, not from sunlight, but rather from energy-rich chemical compounds like H2S, hydrogen sulfide. So there's the spiel. Different strategies for getting energy to drive these metabolic processes. You and I, as heteroautotrophs, are totally dependent upon the autotrophs to keep our metabolic engines running. The reality is that 99.9999% of the energy that keeps our own metabolic processes running is derived from photoautotrophs. In other words, plants that are doing photosynthesis. Okay, so a little bit of pep talk for you, maybe make you appreciate that this is an important topic, because if photosynthesis isn't going on, we have a very short future. All right, so let's get to work and talk about photosynthesis. I warned you there's going to be a lot of green that's up on the screen today, but this slide is pretty much just there to give you the sense that photosynthesis is happening in all sorts of different places. And here's the thing that I think is really quite remarkable. In all of these different types of organisms, cyanobacteria, purple sulfur bacteria, plants, right? In all of them, photosynthesis is playing out exactly the same way. There is one way that living things on this planet photosynthesize. It's common to all of these organisms, despite the fact that they've been diverging and evolving independently for about 1 billion years. All right, so this is another one of those examples of an ancient metabolic process. It must have been hugely important to the very first living things on the planet. They worked it out early on and worked it out in such a way that it's about as good as it can get. And it hasn't been messed with since. There is no new way to photosynthesize that has popped up on this planet in over 1 billion years. There barely are differences in the way that all these different organisms do it. So that means it got worked out early, and it got worked out really well. And I'll tell you, I think it's curious personally, couldn't there have been some other way that this could have worked out? Why can't you have a second kind of photosynthesis going on? I don't have a good answer for that. I think the fact that there isn't a divergent or a new kind of photosynthesis, I think that tells you this must be an awfully good way to get this job done. And the job, again, that we're talking about here is pretty simply, there's energy in them photons, and photosynthesis is in the business of harvesting, of capturing that energy and converting it from electromagnetic energy, which isn't very practical or useful to living things, and instead converting that electromagnetic energy into the energy that we find associated with covalent bonds, specifically the ones stored in sugars. So again, the point here is that all of these different things are doing photosynthesis. They're doing photosynthesis exactly the same way. So what I'm going to tell you is going to apply to all of these different, very different kinds of organisms. Now, for you and I, where we're going to be most interested in how photosynthesis plays out, it's going to be in the context of plants, because we eat a lot more plants than we do cyanobacteria. Right? And so while cyanobacteria, purple algae, seaweed, they're all doing photosynthesis, the types of carbohydrates, the sugars that we're eating, are typically derived from plants. And this slide's a nice one. I think we'll see it again a little later in the lecture. But one more time, I get to try to emphasize to you this idea that structure and function are intimately related concepts in living systems. All right? And it's true at all levels. Here we're talking about it at a microscopic level. It's true at the tissue level, the organ level, the organism level, the ecosystem level, population, no matter what level you're talking about. Structure and function are intimately related. And that is, again, certainly true for the way that photosynthesis takes place. I would be really surprised if you guys hadn't heard before that photosynthesis takes place in chloroplasts, all right? I don't want to insult you and pretend that nobody mentioned that to you in first grade. But more specifically, within the chloroplasts, the photosynthesis is taking place in these, in the thylakoid membranes that are these membranes inside the internal membrane-bound compartment that is the chloroplast. Remember, I made a big deal about how mitochondria, the nucleus, and chloroplasts stand alone from all the other internal membrane-bound compartments inside of eukaryotes, in the sense that they are double membrane-bound. So chloroplasts, according to the endosymbiont theory, were once free-living organisms that got engulfed and became an internal symbiotic organism living inside of some other cell. that eventually became the eukaryotic kind of cells that we know today. Well, here's one other thing that makes the chloroplasts a little bit special. Among those three special double membrane bound compartments, right, the nucleus, the mitochondria, and the chloroplast, among the three, the chloroplast stands alone as having, in addition to that double membrane bound outer wrapping, it has additional internal membrane bound compartments, right? And so this is the only time that we see, I've described a eukaryotic cell as a bag full of goo. Well, it's a bag full of goo that has little small bags. Some of those bags are double wrapped, or double bagged, if you like. One of those double bagged bags has little bags inside of it, right? And it's those membranes inside of it, the thylakoid membranes, which I'll talk with you about a fair bit yet today. That structure is hugely important to being able to get photosynthesis to work. Okay, so we got some pretty pictures here. There's some neat structures that are going on. You can see it's important that there's holes in the surface of a leaf. They are called stomata, through which gases can come and go. Carbon dioxide goes in, oxygen goes out. But the whole purpose of the leaf is to present these chlorophyll bags in such a way that they can absorb light energy and convert that electromagnetic energy into chemical energy. All right, so chloroplasts are where the action are. Before we really get into those structures, let me just take a little while here to talk with you about the chemistry, because there is a basic chemistry that's going on. Here is the overall chemical reaction that's taking place. We're taking carbon dioxide gas, combining it with water. And I would like to encourage you to just pause and take a look at those two structural or chemical formulas. Carbon dioxide gas and water. Think about the structure of carbon dioxide. Carbon with two double bonded oxygens to it. When you look at that structure, I hope now you have been so indoctrinated by what I've been doing to you over the course of the last month or two that you contemplate that structure and you say, wow, that is as low an energy molecule as I think I have ever seen. Right? That's what we're all thinking. I can see all the nodding going on. Yeah, you guys are with it. I've successfully brainwashed you. But I think I've brainwashed you in a good way. This is a really low energy molecule. Those electrons in those double bonds, the carbon, the two oxygens, each with those double bonds, those electrons are hanging out a whole lot closer to the oxygens than they are to the carbons. Okay? So that's like the definition of a polar or of polar covalent bonds. These are low energy bonds. And giving a good run for the money here, water is also a crazily low energy molecule, right? The oxygen is sucking up the electrons. The hydrogens are hanging on for dear life as part of this covalent bond. There's not a lot of sharing of the electrons. Those are low energy electrons as well. So here's the thing. The stuff on the left side of this equation, there's not a high energy bond to be seen. Right? With me on that? And then look what happens. That the cursor arrow here is pointing to an arrow. So that's chemical shorthand for there being a chemical reaction. It's one that's catalyzed by a number of enzymes. The arrow gets a name. That arrow's name is photosynthesis. As you put those two substrates through photosynthesis, look what comes out to the other end. The other end of that function is carbohydrate, right? Carbon with water associated with it. Fancy talk, that's carbohydrate. Carbo for the carbon, hydrate for the water. So you come out with a carbohydrate, and when I say carbohydrate, I'm also thinking that I've brainwashed you into thinking carbohydrate, that's energy, right? There's energy over there. And if you look at that structure, if you contemplate that structure, carbon with a covalent bond to a hydrogen, that's pretty high energy right there. We've got a high energy bond that we're talking about, and then there's also a bond to a hydrogen and an oxygen or a hydroxyl group. Not a lot of energy there, but water soluble. Right? At least you got that going for you. But you got a high energy bond where you didn't have any before, and you're also kicking oxygen out of this process. Okay? You and I think oxygen is pretty cool. It comes in really handy for the types of energy extractions that can happen during photorespiration, the electron transport chain, right? So if you don't have oxygen, you have a hard time getting all the energy out of these carbohydrates later on. So it's neat that we've made oxygen as a byproduct here, because you and I are going to need that when we do this process in reverse. But the oxygen is also a pretty high energy situation. That double bond that connects one oxygen to another oxygen, you have equal sharing of the electrons. There's energy to be had from those oxygen molecules. Again, we'll use that to help drive the process in the other direction. So that arrow, the cursor is pointing to an arrow. That arrow, in a word, is photosynthesis. And that reaction, going from carbon dioxide gas and water to carbohydrate and oxygen gas, it is the exact opposite of what we had talked about with glycolysis in the citric acid cycle, right? If you flipped that arrow around, you're taking the very same substrates to become products in one that are the products that become substrates for the other. It's just it's a yin-yang kind of thing. And you can see now how we're kind of closing the loop. If all that you had going on was this process of photosynthesis, you'd eventually run out of carbon dioxide gas, maybe water, right? And if all you were doing was glycolysis in the citric acid cycle, you'd eventually run out of carbohydrates and maybe oxygen gas, right? The two of them together, we've got a neat, happy system. There's a cycle that's going on. You've probably heard the name of this cycle. It's called the carbon cycle. All right. So the fellow who really cracked this nut and who figured out what's going on with photosynthesis, turns out it was a fairly complicated chemical process. It wasn't as obvious as you might have thought just from looking at that previous equation. The fellow who figured this all out is John Kelvin. And he came up with this clever thing that we now call the Kelvin cycle. And we'll talk more about that a little later in today's lecture. But Kelvin's the guy who figured out how it is that these different atoms end up being divvied up, going from substrate to product, right? And what he found is, in a nutshell, that the oxygen gas doesn't come at all from the carbon dioxide gas. Prior to Kelvin's work, people were just naturally assuming that what you do is you clip the covalent bonds here, kick off the oxygen gas, take your carbon and hook it up to the water, and you're all set. Well, Kelvin's the guy through radioactive isotopes who figured out that the oxygen gas actually comes from the water molecule, not from the carbon dioxide, and that the carbon obviously ends up in the carbohydrate, and that some of the hydrogens come to the carbohydrate, some of the hydrogens go to the water, and the oxygens with the CO2 stay associated with the carbohydrate. So that's the long and the short of what it is that it took Kelvin a career, about 30 years, to work out. And that process, again, is memorializing him. This is the Kelvin cycle. The way that this all plays out is a circular process, just like the citric acid cycle is. Different, but a cycle. In that sense, it's the same thing, where carbon dioxide gas comes in. Note here that we're bringing in 3 carbon dioxide gases, and for each crank of the Kelvin cycle, well, each 3 cranks of the Kelvin cycle, you get 1 glyceraldehyde 3-phosphate, a 3-carbon molecule. That 3-carbon molecule, glyceraldehyde 3-phosphate, is one whose name you may remember from lecture on Tuesday and Thursday of last week. because it's part of the citric acid cycle as well. So it's a good straight shot back into the citric acid cycle and run this in a different direction. We'll talk more about what's going on in this Calvin cycle, but I want to just toss it out to you right now so you get a sense for the general lay of the land. Before we talk about that chemistry, let's talk about really the first step in the process. I'll steal some of my thunder, and I'll tell you right out of the gate here. There's two parts to photosynthesis. There are what are called the light reactions, and then there are what are called the dark reactions. All the chemistry of the Calvin cycle that I just flashed at you, those are the dark reactions. And they're called the dark reactions because, it's kind of clever, they can take place in the dark, right? In the absence of light. Okay? The light reactions, in contrast, are all about light. They only take place in the presence of light. And you can't do the dark reactions unless the light reactions happened first. So what often, it's a little bit of an oversimplification, but you could suggest that during the daytime, plants are busy doing all the light reactions, building up a bunch of energy-rich molecules that they then use to drive the dark reactions at night. The reality is the dark reactions are kind of happening all the time, but the light reactions, because they are the light reactions, only take place while there's sunlight to be had. So here's what's up with the light reactions. This is the start of the whole process of photosynthesis. It's the first half of photosynthesis, the light reactions. What we got to do is we need to, again, capture the electromagnetic energy associated with photons and store it in something that's more permanent, more tangible, that's something that you can transfer. We want it in covalent bonds. We want to store it in energy-rich electrons. So the first step in this process has to be the absorption of those photons, of those high energy, of the photons that have a lot of electromagnetic energy, so that we can then do things with that energy in the sense of making some covalent bonds. So in the light reactions, light gets absorbed and water gets utilized, and we're also taking in these relatively energy poor molecules. You'll probably remember NADH and NAD from the lecture on Tuesday and Thursday. Well, here's a subtle modification of NAD. It's NAD that's been phosphorylated, NADP. So we're taking in this low energy molecule, and you guys all know, I'm sure by now, ADP is relatively low energy, and inorganic phosphates are seriously low energy. We take these low energy molecules, we use some water, we absorb energy from light, and we end up cranking out oxygen gas. And look at these happy, bright, shiny, energy-rich molecules. ATP, right? Synonymous with energy for living things, used to drive just about all metabolic processes. And NADPH, which again is the rough equivalent. It's not even a rough equivalent. It is the equivalent of NADH, right? The same molecule, now phosphorylated, subtle difference between what we had been talking about all of last week. So here are the light reactions in a nutshell. Take these energy poor molecules, including water, juice them up with some energy from light, release oxygen gas, and make these energy-rich molecules. And what do you suppose takes place on the other side of this screen? Here are the light reactions of photosynthesis. In a moment, you're going to see what happens with the dark reactions. Those are the dark reactions of photosynthesis. The dark reactions of photosynthesis take in carbon dioxide gas and use these energy-rich ATPs and NADPHs that were created during the light reaction, or the light reactions, to drive this sort of pump, this cycle, keep that wheel moving. Carbon dioxide gas comes in, these energy-rich molecules come in, energy-poor molecules come out, but can be recycled and reused and run this process one more time. And perhaps most importantly of all, carbohydrates come out. In other words, sugars come out. You can look at, this is a cool picture, guys. You may not think this cartoon is a particularly good cartoon at the first pass, but boy, this cartoon, it's got a lot of stuff going on, diagramming what's going on with this process. One of the things that makes it cool is I think you can look at it diagonally too, right? We're putting light energy in, and we're coming out at the other end with energy stored in covalent bonds. Notice also that there's a cycle that's going on here. There's an ADP, NADP, ATP, NADPH cycle that's churning away, right? Notice where this is taking place. This is taking place in this thing that we call a chloroplast, right? None of this stuff is taking place in the cytoplasm. Remember with the glycolysis and the citric acid cycle, glycolysis happens exclusively in the cytoplasm. The citric acid cycle for you and I happens exclusively in the mitochondria. Here, both of these reactions, the light and the dark reactions, are taking place exclusively in the chloroplasts. Notice that the light reactions are taking place exclusively in the thylakoid membranes, these stacks of membranes inside of the chloroplasts. And notice that the Kelvin cycle, the dark reactions, if you like, are taking place not inside the thylakoids, but rather in the space that's inside the chloroplast that isn't the thylakoids. So again, there's a lot of stuff happening in this particular cartoon. Right? But in a nutshell, it's giving you a roadmap of all the things that are going to be taking place that together we call photosynthesis. Right? So there's that arrow expanded for you a little bit. This is photosynthesis. On the left side of the screen are the light reactions. On the right side of the screen are the dark reactions. Now, I'm pretty sure I'm certain that what I've just talked with you about here is not the first exposure you have had to photosynthesis, right? But I think we've probably gone a little deeper than most of you have thought about for photosynthesis in the past, all right? So it may be that your first exposure to photosynthesis involved something like this. For those of you who know, you can explain to the others what's going on there. I'm sorely tempted to imitate for you all SpongeBob's interpretation of the photosynthetic process, but I'll let you imagine how that might have played out. Some of you know, I can tell. All right, so photosynthesis. We've seen a look now at this overall process. You know, I hate to say it, but this is another one of those redox processes. The key here is this. Again, we're keeping our eye on where it is electrons are going, and particularly how much energy is associated with those electrons. Are these electrons high energy electrons because they're far removed from the atomic nuclei that they're associated with? Or are they low energy electrons because they're close to an atomic nucleus, right? It's a redox reaction in the same sense that these other reactions have been redox reactions. We're looking to see where electrons are moving, and specifically we're looking to see how the energy status of these electrons change. I don't really want to say anything more about the redox process than that. So let's get to work and talk about these light reactions. I've said it 3 or 4 times now already today. This is all about harvesting energy stored in photons. It's all about harvesting and converting electromagnetic energy and converting it into chemical energy. It's not a simple trick. It's not something that you and I are capable of doing at all. Right? And so to talk about what's going on with these light reactions, let's take just a minute to explore how photons could be considered to have energy in the first place. Right? So photons are these particles of sorts that's got a dual nature. You've probably heard before. It's simultaneously a wave, but it also has behavior as if it is a particle, right? So photons are these sort of fuzzy things that are hard for us to wrap our heads around. They act like a wave, but they also act like a particle at the same time. For our purposes, we don't need to grapple with that sort of simultaneously inconsistent way of thinking about something. That's not our concern for this class. For our purposes, it all comes down to how much energy is in these photons. And how much energy is in a photon boils down to one thing and only one thing. It's the wavelength of the light that you're talking about. So again, photons have this dual nature, wave and particle. The wavelength is the distance between one peak on a wave to the next peak on a wave. Right? And a lot of times you'll see photons shown as sort of a sine wave, right? So from one peak to the next peak, or from one trough to the next trough, the distance between those two peaks or those two troughs is not an assassination. It's not an assassination attempt. Don't worry. So it's the distance between the peaks or the troughs, right? That distance is the wavelength. And it all comes down to this. The shorter the wavelength, the shorter the distance between those two peaks on the wave, the greater the energy, right? These photons pack more punch if they're on a short wavelength as opposed to a long wavelength. And the word spectrum turns out to be crazily appropriate to use in this context. This is the original context for the word spectrum. The spectrum of wavelengths, the range of different wavelengths that we know can be associated with photons, covers pretty much the whole gamut. The really tiny wavelengths, the ones with lots and lots of energy associated with them, are the gamma rays. Next up, in terms of the wavelength distance, the separation between those peaks, are the X-rays. Then we start to move into this part of the spectrum, the ultraviolet part of the spectrum. You get this little tiny narrow range of about a few hundred to about 1,000 nanometers for the wavelengths that we associate with visible light. And then you move out of that. When the wavelengths get a little bit bigger, you get into the infrared, all about heat, right? And then bigger still, longer wavelengths still, we're starting to talk about microwaves. And longer wavelengths still, we're in the radio wave part of the spectrum. So, maybe it'll help you appreciate this. If I give you the choice of being inundated, I'm going to shoot a beam of photons at you. What kind of photons do you want me to throw at you? Do you want me to throw at you gamma rays or radio waves? Nope, nobody wants to get hit with gamma waves, right? It's a bad idea. Gamma waves would hurt you, okay? We're actually being saturated. We're constantly, radio waves are all over. You guys have cell phones that are emitting radio waves. You're soaking me up with radio waves right now. And I don't know, I don't care, it doesn't bother to me at all. In fact, text away, have a lot of fun, tell people you're using radio waves to transmit signals. I don't care, it doesn't matter to me at all. Radio waves are photons. It's just the wavelength there is on the order of a kilometer, right? And there's such long wavelengths that these photons barely pack a punch at all. The shorter the wavelength, the more energy associated with it. Nobody wants to get attacked by gamma rays. I think you all know you want to limit your exposure to X-rays because those photons can mess you up too, not as quickly and as badly as gamma rays. UV light, that could cause you some problems, sunburn sorts of things. But visible light doesn't hurt us that much, and by the time you're up to the infrared and the microwaves, those things barely pack a punch at all. But again, we want to focus our attention here on what's going on in the visible light, because these are the wavelengths that chlorophyll, the thing that makes chloroplasts green, chlorophyll is really good at absorbing visible light, right? Kind of gives a pass to UV, gives a pass to infrared, all the other parts of the spectrum. Chlorophyll is primarily about absorbing visible light. And interestingly, chlorophyll does a bang-up job of absorbing all of this light from 380 nanometers up to about 530 nanometers. It does a really good job of absorbing light in that wavelength, photons that have that wavelength. And it also does a really good job from about 600 to about 750 nanometer wavelength light. Chlorophyll sucks that stuff up. One of those photons touches a chlorophyll, and the chlorophyll absorbs it and collects its energy. So chlorophyll is great at absorbing light in those two parts of the visible spectrum. Curiously, it's not so good at absorbing green light, right? And that's why chlorophyll looks green. With me on that? Everything else gets sucked up. White light is everything in the visible spectrum. Right? So when we're talking about white light, it's not a color, it's actually all colors. Black, in contrast, is the absence of light. White light is a mix of photons in all wavelengths. Green light is photons, but of a particular range of wavelengths. And so chlorophyll does this great job of absorbing light on the low end of the wavelengths for the visible spectrum and on the high end of the wavelengths for the visible spectrum, but it doesn't absorb the photons that are in the green part of the spectrum. Those bounce off, those get reflected off and can hit our eyeballs, and our eyeballs then perceive, look, there's something that has a lot of green photons associated with it over there. All the other photons in white light have been absorbed. The green ones are all that are left. That's why we perceive green. And again, this is all because of the way that chlorophyll works. So here's another way to talk about it. I did it with words before. Here's a picture that conveys that same story. Light here from our sun comes from the sun as white light that we perceive, and that white light, which has all of the different photons with the different wavelengths, ends up hitting the chlorophyll in the thylakoid membranes, gets absorbed, and only the green ones, only the green photons make their way through, either as transmitted light or as reflected light. That's why plants look green. And so again, it's all about chlorophyll. That's what chlorophyll looks like as a chemical structure. You don't think you need to memorize that, do you? I hope you don't. You should know me better than that by now. I'm not going to make you have to sketch out that structure. But when you look at it, if somebody flashes it at you on a test or just, here, what's this? What do you think about this molecule? I hope some things jump out at you about this molecule. I think the green stuff at the top is probably where your eye gets drawn to first. So let's get that out of the way. That's where the pigment part of chlorophyll is. All of those ring structures that are in there, ring structures have a tendency to absorb light. Right? So cytochromes that came up briefly in the lecture on Thursday, they have colors associated with them. Their colors are there because they have a lot of these ring structures as well. Most of the pigments and dyes for clothing and food coloring, we're talking about carbon molecules that have these sort of funky ring structures associated with them. There's light absorbing capacity in that kind of structure. And so this is where it's a porphyrin ring. This is where it is that the photons are going to end up getting absorbed. But I want to make sure you guys notice this stuff here, which is a little more subtle. It doesn't have the big green circle around it. Check this out. Each of those kinks is supposed to represent a carbon. And at each of those kinks, there's not just a carbon, but there would also be hydrogens on either side that round out the structure. What are we talking about here? That is a hydrocarbon tail. Hydrogen, hydrocarbon, carbon. It's a hydrocarbon tail. What does hydrocarbon mean to you guys? Again, I've brainwashed you effectively, I hope, and that is this is going to be lipid soluble. Hydrophobic, afraid of water, avoiding water. Instead, where's this going to be? Where's that part of the molecule going to be? That part of the molecule is only going to be happy if it can be embedded in a membrane. Okay, so chlorophyll is a molecule that has that porphyrin ring on one end that's kind of hydrophilic, that's sort of kind of hydrophobic. I don't know, it's kind of a mishmash. But it has this very intensely hydrophobic portion that anchors it in a membrane. And in what membrane does it anchor? It gets anchored in the thylakoid membrane, that bag within the double bag that is the chloroplast. That's where chlorophyll hangs out. This molecule doesn't get embedded in the membrane of the cell's membrane on the outside. It's not the nuclear membrane. It's not even in the chloroplast membrane or membranes. It is exclusively in the thylakoid membrane. Okay? So if I were to ask you, what part of a plant's cell, which of the following membranes would have a green color, would be a reasonable way to phrase it. How about a nuclear membrane? No. Why would you think that? How about a mitochondrial membrane? Inner mitochondrial membrane? No. Outer mitochondrial membrane? No. Lysosome? Golgi apparatus? No. No. Right? Chloroplast? Internal membrane? No. Chloroplast external membrane? No. Thylakoid membrane? Big yes. That's the only thing that would look green, because thylakoid membranes have chlorophyll embedded in them. They're hanging out. Chlorophyll's hanging out there because of this hydrocarbon tail. It's very hydrophobic hydrocarbon tail. All right. So we've talked a little bit about electromagnetic energy. It's related to the wavelength for the photons that we're talking about. The shorter the wavelength, the more the energy. Now it's time to talk a little bit about the photooxidation of chlorophyll. Okay, because this is where the rubber's hitting the road. This is where we're going to be capturing electromagnetic energy and converting it into some chemical bonds. Here in one picture is photooxidation of chlorophyll. All right? And so what's happening is this. Photons with their energy, isn't that cool? They're trying to make you think of a wavelength here, I think. All right? So we have photons of a particular wavelength get absorbed by chlorophyll, this green thing in the membrane that is the thylakoid membrane of a chloroplast. So photons get absorbed. That electromagnetic energy from the photon, when it gets absorbed by the porphyrin ring, starts exciting the electrons in that porphyrin ring. That's kind of a key step for the whole process. We took something that's sort of ephemeral, nebulous, this photon, this electromagnetic energy, and its absorption by the porphyrin ring excites an electron. It gets an electron moving faster, maybe more importantly, moving further from an atomic nucleus. We've gone from a low energy electron now to a high energy electron as a result of that absorption of the electromagnetic energy. That high energy electron, we have used a photon to oxidize that electron. So this is a photooxidation of chlorophyll. That high energy electron will typically get passed off to another porphyrin ring for another chlorophyll, and another, and another, and another, often many times before it ultimately gets transferred to some special chlorophyll molecules that are going to be accepting that high energy electron. That high energy electron then, once it's made it to this special set of chlorophyll molecules, gets passed off to a reaction center, and the reaction center then does the rest of these light reactions of photosynthesis, which will ultimately convert NADP to NADPH and ADP in inorganic phosphate to ATP. Okay? First step in the process is this photooxidation of chlorophyll. You excite an electron, a low energy electron becomes a high energy electron, and now you got something that you can hand off to this reaction center complex to do the rest of the light reactions. So here are the rest of those light reactions. Well, actually, first step, we absorb the photon, pass it around a little bit, get it to these special chlorophyll molecules that are linked to the reaction center, hand off that high energy electron to a primary acceptor, then pass it through a chain where you can do substrate level phosphorylation. Okay? We've seen this kind of electron transport chain before in the context of what's going on in the mitochondria, the same idea. And ultimately, that high energy electron gets passed down the chain. That electron transport chain turns a crank, smushes together an ADP in an inorganic phosphate to make an ATP, but it's still high energy and can then get passed off again after some more light comes along to excite it. That electron and another electron can get together to this pair of chlorophylls again and go back to the primary acceptor, and you can drive that process one more time through an electron transport chain that ultimately drops off the electron to its original low energy state, but only after it converts an NADP plus to an NADPH. So, one photon getting absorbed gets us an ATP. A second high energy electron or a second photon getting absorbed can jack up that electron again to an even higher level and bring us to a point where we can make NADPH. 2 photons, and we end up with 2 high energy molecules that we didn't have before. These are the light reactions of photosynthesis. Because again, none of this happens unless you have photons that are jacking up those electrons to get the process underway. You could think of it this way, if you like. Another cartoon that gives you a sense of what's going on. We've got a low energy electron here that gets jacked up because a photon is really giving it a boost. That high energy electron moving around at a high level because a photon has jacked it up can be passed down through this system here, an electron transport chain that turns a crank that produces ATP from ADP and inorganic phosphate. That electron, jacked up still, it's not completely back down to the level it was before, but that electron can get excited by another photon, which jacks it up to an even higher level, and that in turn can be used to drive the creation of NADPH. Okay? So we have this process of photooxidation taking place. There's cycles going on here. We don't need to say too much more about that. There are 2 photosystems, photosystem 1 and photosystem 2. Right? Here's photosystem 2, that's where it starts, inconveniently named. And then photosystem 1 is what finishes off the process for you. So when we talk about photosystem 1 and photosystem 2, that's what this is all about. So there are 2 photosystems, and notice that the electrons are kind of flowing through this whole cycle. All right. Well, let's talk nuts and bolts about how it is that these high energy electrons and the electron transport chains are being used to create ATPs and NADPHs. Okay? Does this look at all familiar to you? I hope it's vaguely reminiscent to you of what it is that we were talking about for what's happening in the mitochondria with the photorespiration, the oxidative phosphorylation that we talked about on Thursday of last week. It's got the same sort of look and feel to it. It's sort of set up in the same sort of way. Check out what's going on here. We've got, there's something that's hugely different but there's also an awful lot that is the same as what I talked with you about on Thursday. So check out what's going on here. We've already talked about this stuff. The light, the photons get absorbed by the chlorophyll, gets handed around, right? Goes through an electron transport chain. As it goes through an electron transport chain, notice what happens here. We're taking hydrogen ions from one side of a membrane and pumping them in to another side of a membrane. You know what this membrane is? This membrane is the thylakoid membrane. Okay, that's where chlorophyll is. Right? Those little green spots are chlorophyll. This is green in color, though it's not shown in green right now, but it would be green because it's got a lot of chlorophyll there. So, picture where we're at. Just want to remind you, you got a double membrane bound thing that we call a chloroplast. Inside the chloroplast are additional membrane-bound compartments. Those additional membrane-bound compartments are the thylakoids. The space inside of a thylakoid, the watery stuff inside, that's called the stroma. Okay? And what's happening during the light reactions of photosynthesis is this. Photons are absorbed by chlorophyll. Those high-energy electrons are passed through an electron transport chain. As they pass through an electron transport chain, just as was taking place in the mitochondria, the electron, as it goes from high energy down to low energy, turns some cranks and facilitates the movement of 4 hydrogen ions across the thylakoid membrane into the stroma. More electrons get excited, and, well, photosystem 1, so light gets excited here, and then the NADPH gets made here on the outside of the process. But check out what's going on here for these hydrogen ions with photosystem 2. Each photon that excites an electron in photosystem 2 is going to result ultimately in the movement of 4 hydrogen ions from the chloroplast space from the stroma, which is the outside of the thylakoids. I misspoke before. It moves it from the goo inside of the chloroplast, the stroma, to what's inside the thylakoids, the thylakoid space. All right, so just as in the mitochondria, what's going on here with these excited electrons being passed through an electron transport chain is we're setting up a hydrogen ion gradient. Okay, there's going to be a different pH inside the thylakoid space than in the stroma, the stuff outside of the thylakoid. And just as was the case for the mitochondria, nature doesn't like this. It doesn't like the separation of charge. It doesn't like that there's more positively charged things inside the thylakoid space than outside. It doesn't like the difference in the concentration of the hydrogens, right? More inside the thylakoid space than outside. And living things have worked out this deal where they say to nature, I know you don't like this setup. I'm going to help you out. I'm going to let some of those protons get back across the membrane, but I only will do that for you if you let me turn a crank that lets me create ATP from ADP and inorganic phosphate. What's the protein that lets you do that in mitochondria? It has a name. I mentioned it. It's ATP synthase. Right? What's the protein that lets you do that in a chloroplast? It has a name. It's the same protein, ATP synthase, and it works the same way. It's a gate that lets these hydrogen ions go from high concentration to low concentration, down a diffusion gradient, and as they're flowing through, a crank gets turned and smashed together in ADP and an inorganic phosphate, the same as it was happening inside the mitochondria. So a whole bunch of similarities. In fact, the protein is the same protein, ATP synthase. It's the same as the one that's working inside the mitochondria. There is a difference. The difference that we're seeing here with chloroplasts and photosynthesis than what we saw with the going on and what's going on inside the mitochondria is the direction of the flow of the protons. In the mitochondria, the electron transport chain's orientation is in the other direction. What we're doing in the mitochondria is we're taking electrons, or we're taking protons from inside the mitochondrial's inner space and pumping them to the outside, and then letting them flow through in the other direction. So the orientation here is different, but all the other players are the same. Kind of cool. Worked something out well about 1 billion years ago, and we've played with it ever since. So here you get a side-by-side comparison of what's taking place in the mitochondria relative to the chloroplasts. The gray area, excuse me, the gray spaces here are supposed to make you think that's where it is we've pumped protons into. And the lighter colored spaces are where we're allowing the protons to flow back. So you could think of the coloration here being an indication of how many protons are there. Squint your eyes. That gray, the darker gray is where there are a lot of protons. The lighter gray is where there aren't as many protons. Again, the orientation in the mitochondria is different than what it is inside of the chloroplast, but the protein that acts as the gate that lets you synthesize the ATPs is one and the same. Okay? So, electrons are flowing. ATPs are getting made by ATP synthase, and ADPH is being made at the same time. Let me just go back here and point out one other thing to you. Notice where the NADPH is. It's on the outside of the thylakoid. Notice where the ATP is. It's on the outside of the thylakoid. Convenient, because where it is that the dark reactions of photosynthesis are going to be taking place is in the stroma, this gray area here, not inside the thylakoid space. And where are the ATPs and the NADPHs? In the stroma, not inside the thylakoid space. All right. So, photosynthesis, that's what this is all about. Guess what? It's time to talk finally, and I mean finally in the sense that this is the last thing I really need to talk with you about today. Let's talk about the Calvin cycle. In other words, let's talk about the dark reactions of photosynthesis. So it's a cycle, Calvin cycle. We're going to be taking some things through a circle, a circular process. The first step in the Calvin cycle is arguably the most important. A lot of people would tell you unequivocally this is the most important step in the Calvin cycle. What makes it so important to people is this is where carbon fixation occurs. Carbon fixation, interesting sort of idea. What it means ultimately is we're taking carbon, which is in this floating around gas, bopping around carefree, devil-may-care carbon in the atmosphere. We're taking those kind of carbons, carbon dioxide, and we're anchoring them down. We're fixing them. We're attaching them to other carbon molecules in a way that anchors them down. We're taking gas and we're making it into a solid, a sugar. All right, so carbon is getting fixated. It's getting fixed through this process. The very beginning of the Calvin cycle is where carbon fixation occurs. And what that means, in other words, is we're taking a carbon dioxide gas molecule and we're hooking it up to something else. Check it out. It is a 1, 2, 3, 4, 5 carbon sugar called ribulose bisphosphate. 1 carbon plus 5 carbons gives you 6 carbons, which quickly break apart into 2, 3 carbon things. Carbon got fixated. There's an enzyme that catalyzes this reaction. That enzyme has a name. This is like, your name is Robert, mind if we call you Bob? It's one of those kind of things. This enzyme gets talked about so much that nobody ever bothers calling it by its formal, 55-letter long name. Everybody just calls that enzyme Rubisco. Okay? Rubisco, by many accounts, is the single most abundant protein on this planet. Right? No hyperbole, no exaggeration. There are a lot of people who will tell you there is no protein that is more common on this planet than Rubisco. That should tell you something, right? This is a really important protein to living things as we know them on this planet. Rubisco does this really important job of carbon fixation. And all of photosynthesis is dependent upon it. And by inference, then, we are totally, as heterotrophs, are totally dependent upon the health and well-being of this enzyme as it appears in plants as well. Rubisco, again, is an enzyme. It's a protein. It catalyzes one reaction. It combines ribulose bisphosphate to carbon dioxide gas to give rise to a 6-carbon molecule. What do you guys need to commit to memory? Well, just because so many people are so enamored with Rubisco, I hope you'll remember that's the name of this enzyme. So here's an enzyme name that you probably should remember. Rubisco. It's the first step in the Calvin cycle. It is the enzyme that's responsible for carbon fixation. Rubisco catalyzes a reaction that involves a 5-carbon sugar. It might be good for you to remember that sugar's name is ribulose bisphosphate. The 1-carbon thing, I sure hope you know the name of, that's carbon dioxide. Right? When you take a 1-carbon thing and you hook it up to a 5-carbon thing, you get a 6-carbon thing. The 6-carbon thing becomes 3-phosphoglycerate. Okay? Then, we take energy-rich molecules like ATP and convert them to ADP. An enzyme does it. They're not even bothering to scare you with the name of this enzyme. They figure you can't handle it. You can look it up. It's not that scary. You know what it is. I tell you what, I can quiz you right now. What do you think it is? What's going to be part of the name of this enzyme? Is it going to be a kinase, a dehydrogenase, or an isomerase? It's a kinase, right? There's a phosphate group that's being transferred, right? So a kinase does its thing and converts 3-phosphoglycerate to 1,3-bisphosphoglycerate. Only difference is now there are 2 phosphates where there used to be just 1. If I showed you this molecule and this molecule, you'd probably have a pretty easy time of telling me there's more energy stored in this molecule than in this one. Be right. Okay? Then another enzyme does its thing. Part of the name of this enzyme is it's a dehydrogenase because we're transferring a hydrogen from NADPH to NADP. And we end up then with glyceraldehyde 3-phosphate. If you crank this wheel 3 times, you have the opportunity to shunt away at this point glyceraldehyde, 1-glyceraldehyde 3-phosphate molecule. That's a carbohydrate, and that's something you can plug right into the citric acid cycle and use to make carbohydrates, bigger sugars, things like that. Okay? But to complete the cycle, we need to take those glyceraldehyde 3-phosphates that did not get shunted off, and get them ready to begin the process again. Some other kinase needs to kick into gear for us, and we're back to having ribulose bisphosphate. You can see they've done some shorthand here. There's actually 3 enzymes that are involved with this process, but you make this substrate again, the ribulose bisphosphate, and you're ready to begin the process one more time by feeding that to Rubisco and letting Rubisco then fix some more carbon dioxide gas. You've got this cycle going. Each 3 turns of the cycle, you end up being able to make 1 new glyceraldehyde 3-phosphate. A molecule that's a sugar that has a lot of electrons stored in high energy state. So those are the dark reactions of photosynthesis. Let me touch on one more sort of topic with you here today. I think we'll be finishing a little bit early. There's such a thing as a C3 plant and a C4 plant. Okay. C3 plants are ones that do carbon fixation in the way that we've just talked about here. They rely upon rubisco to do their carbon fixation. For as wondrous an enzyme as rubisco is, abundant everywhere, it's got a serious Achilles heel. It's got a serious flaw with its design and the way that it works. You would have thought this would have gotten worked out. The fact that it hasn't been worked out over like a billion years means it must be a really tough problem to solve. But here's rubisco's big problem. Rubisco can sometimes get confused by oxygen gas and use oxygen gas in place of carbon dioxide gas. If you imagine what those structures look like, the ends of carbon dioxide, it's the same shape of a molecule, it's just a little bit longer, right, than oxygen gas. If rubisco is exposed to oxygen gas at the same time that it's got ribulose bisphosphate around, it can attach the oxygen gas to the ribulose bisphosphate instead of the carbon dioxide. It can make a pretty serious mistake. When that happens, here's a word I want to make sure I get right for you here. When that happens, it's photorespiration. Oh, see, tells you how much I'm going to expect you guys to remember this for a test. I can't even dredge it up out of my notes. No, it's not this word. Yeah, now this is getting embarrassing. All right, so I'm not even going to take a stab at what this word is. I'll look it up, and I'll tell you what it is when we get back together on Thursday. But here's the thing. When oxygen gas gets hooked up to ribulose bisphosphate, ribulose bisphosphate, when oxygen gas gets hooked up there, the ribulose bisphosphate is taken out of the game. It's done. It's terminal. You can't recover from that. When rubisco makes that mistake, it takes out a ribulose bisphosphate from the cycle. Ribulose bisphosphate is a very energy-rich molecule. This is no small deal. This is huge, right? That mistake is a very costly mistake for a cell. It's a very wasteful step for a cell. To be able to recover from it, you have to be able to do the job right at least 5 times for every time you make the mistake, or you're running this Kelvin cycle at a loss. That's not what this photosynthesis is about. It's not about getting rid of energy-rich molecules. It's about building them up. Right? So when rubisco makes that mistake, it has some pretty serious consequences for a cell. For plants that are C3 plants, what that means is that they are only capable of doing carbon fixation with rubisco. Okay? C3 because the first molecule in which the carbon gets fixed ends up becoming glyceraldehyde 3-phosphate, a 3-carbon molecule. That's why it's a C3 plant. Plants that can only do carbon fixation with this Kelvin cycle are at a disadvantage to plants that can do it some other way that allows you to avoid that problem that rubisco has. So there are plants that we see all the time out in fields and such that are C3 plants. There are other plants that are C4 plants. The C3 plants are the ones with the broad, flat leaves, soybeans, squash, pumpkin vines, trees. These are C3 plants. That's all that they can do is work with rubisco. Those guys have a serious problem when oxygen concentrations are high and carbon dioxide concentrations are low. You know when that happens? That happens on hot, dry days when the plant's stomata, those little pores on the surface of their leaves, close up to be able to conserve water, to save water, so you don't evaporate away a lot more of that water, because it's a hot, dry day. Carbon dioxide then inside of the leaf goes down in concentration because it's getting fixed through carbon fixation with rubisco. And remember, oxygen gas is a byproduct, it's a waste product of this whole process, and carbon and oxygen levels start to go up. But it can't get out of the leaf because the stomata have closed. Carbon dioxide can't get in because the stomata has closed. On hot, dry days, leafy plants have an issue. They've got a problem. They're just going to have to put the brakes on the whole thing, and they're missing an opportunity to fix carbon, to capture energy. They're at a disadvantage over plants that have an alternative. The plants that have an alternative are the C4 plants. Those are the grassy kind of things, like, oh, let me think, grass, all right, or corn, all right? There's another C4 plant for you, all right? Those types of plants, sugarcane is another one. Those types of plants have solved this problem in an interesting way. They do their carbon fixation not with rubisco, but rather with PEP carboxylase. And so they have this one extra cycle that they can bring to bear. It costs them an ATP to do this, but it allows them to make a carbon molecule, fixate carbon to store some energy in a high energy bond that it can then break down later on and drive into the Calvin cycle. When oxygen levels are low, the carbon dioxide is high. All right? So these C4 plants, instead of fixing carbon in a 3-carbon molecule right out of the gate, it's pretty much their only choice. C4 plants can do both. They can sometimes capture it as part of a 4-carbon molecule and then ultimately convert it into 3-carbon molecules and get the same job done. So C4 plants can run both ways. They can run both on a C4 sort of cycle and a C3 kind of cycle, whereas C3 plants just can't. All right, one more time, pretty picture for you here. The light reactions of photosynthesis take place in the thylakoid membranes. Those get used to make ATP and NADPH, and they release oxygen gas. It's a byproduct, an end product, a waste product, if you will. Those energy-rich molecules can crank the Calvin cycle, which ultimately results in carbon fixation and releases these energy-poor molecules. And the carbon, as it gets fixated, can end up being used to make sugars like sucrose. That's what I got for you here today. We got 2 things I should mention as you think about this question that'll be on your next test. One is you guys are in for a treat in lab this week. You get to play with microscopes, so don't break them. They're expensive. But you get to look at some cells, and you get to look at the stuff that I'm going to be talking with you about on lecture on Thursday, cell division sorts of things. But here's one of the 2 things that I think is really cool. You guys know how crappy the chairs are in the lab, right? No more. Brand new chairs in lab for you this week. How about that? I pulled some strings, and you guys have nothing but fancy new chairs. They go up and down, they swivel around, they have backs. You're going to love it, all right? And the other thing is when I see you guys on Thursday, we're going to talk finally about cell division sorts of things. All right, see you guys later.